The title of my talk, What is Life?, is of course a homage to Schrödinger. It is really, in my view, the most fundamental question in biology, because the answer to that question defines the biosphere, the distinction between the living and the non-living worlds. And a discussion of the question illuminates, again in my view, some of the most important properties and characteristics of life. And that's what I want to do this evening, to try and indicate what the differences are between the living and non-living worlds, and also discuss, as a consequence of that, characteristics of life. As we've heard, it's the title of the book by Schrödinger, which was given in Dublin and published in the mid-1940s. Schrödinger, of course, was one of the great physicists, in fact, one of the great intellectuals of the 20th century. Austrian, born in Vienna, though a quarter English, one of the fathers of quantum mechanics, and of course also the father of Schrödinger's cat, which many know him because of, and also derived the Schrödinger equation, which describes wave mechanics, forced to leave Austria in the 1930s and went to Ireland. And it was in Dublin, as we've just heard, in 1943, that he gave the lectures on What is Life, which he then published in this lovely little book here in 1944. It's a book I read myself in the 1960s. I have not quite a first edition, but nearly a first edition. And this book focuses particularly on one characteristic of life, which I will also discuss, and that's heredity. He argued quite rightly that heredity was a key characteristic of life, and also argued that heredity, which was based on information, required substances in the cell that had chemical stability. There's a number of quotes in his book, and I've chosen two which identify that. He coined the term code script as a term to describe information. And the first of the quotes here, these chromosomes, that is the carriers of the hereditary information, contain in some kind of code script the entire pattern of the individual's future development and functioning. The notion being that the chromosomes had a code script that codes information, and the entire pattern of what happened to a living organism was coded within that code script. And then he thought and discussed in his book that for that to be passed on from generation to generation, it had to be extremely stable. So he thought about the chemistry behind it and came to the conclusion it is solidity on which we draw to account for the permanence of the gene. So we believe a gene to be an aperiodic solid. And he was thinking of crystals. He's thinking of an aperiodic solid in describing that. We know it's not like that, but the notion of chemical stability is absolutely implicit there. And this makes up the reasoning behind these two statements here, makes up the majority of that little book. But he also mentions, though rather more briefly, another aspect of life, which I shall also talk about, and that is the question of self-order, the ability of an organism to self-organize. And as a physicist, he took the position that you see here. He was thinking of chaos and order and the second law of thermodynamics. And two quotes again, an organism's astonishing gift of controlling a stream of order on itself and thus escaping the decay into atomic chaos. Existing order displays the power of maintaining itself and producing orderly events. So this is emphasizing order, self-organization, self-maintenance, all of which, again, I shall touch upon in tonight's talk as well. Now, this book had a great deal of influence on early molecular biology. It influenced particularly Francis Crick. Some of the arguments in there had been made by others. I'll point out one or two of those during my talk. But the key point that information is implicit in the concept of the gene, he emphasized, and the need for chemical stability. So that was extremely important for him. But the book is beautifully written, it's beautifully argued, and I think the fact that an outstanding theoretical physicist thought that biology was worth writing about in the 1940s was also an important point, quite frankly. At the time, and even to the time when I was at school, biology was often seen as the second-class science, you know, for those that were bad at maths and quite good at drawing. And I suppose that's the category I was put in and sort of put into the biology stream. But the fact that a great mind like Schrödinger could actually divert some of his attention to this great issue, I think made the subject seem important, and I think inspired those who were starting the molecular biology revolution a few years later in the 1950s, who often themselves came from a physics background. So I think it played a role in making biology, taking biology seriously. Now, tonight, of course, I'm going to ask the same question as he did. And I'm going to do it by discussing the attributes of life. How you can recognize life when you see it. It's rather like the American High Court judge talking about pornography. Difficult to define, but you always know when you see it. And it's the same issue, in fact, with defining life, because it's actually quite difficult to give a single answer, a single definition. If you want a single answer to the ultimate question of life, then of course the answer is 42, and I don't have to say anything more about that. That's Douglas Adams, if some of you are too old to know what I'm talking about. So I will begin with what life is made of, or at least what life is made of as we know life to be on planet Earth. Now, living things are entities. They're bounded objects. I just want to emphasize that because I'll touch upon this a little later when talking about thermodynamics. They're bounded objects separated from the environment, and they are built on particular chemistries. And there's one aspect of that chemistry which is very important, and that's carbon. Now, the fact that life is built on chemistry was not actually always obvious. Aristotle, for example, argued that life had a teleological purpose, that it was imbued with vitalism, which was different from the laws of physics and chemistry. And it took quite a long time to show that Aristotle wasn't correct, well into the 19th century. And the fact that life is built on chemistry, central to that idea is that chemical transformations occur during the fermentation of fruit. And why that's important is that this was associated with biological function, and therefore scientists began to think that chemistry was critical for life. This had its origins in Antoine Lavoisier, the father of chemistry. And Lavoisier showed that during fermentation, the sugar of the grape was changed to alcohol. And he concluded the following thing, that vital processes of life are chemical reactions. So it was a critical point. He concluded that in the late 18th century. This is a great painting. It used to belong to Rockefeller University, of which I was president until earlier this year. They had to sell it during one of their periodic financial crises, as we all do, and it was sold ultimately to the Metropolitan Museum of Art. It's by David. You can see it there. It's worth $100 million, I have to say. This is his lab assistant here. And she was his lab assistant, but she also was his wife. And later on, she married actually Count Rumford, so another famous scientist. Lavoisier, unfortunately, didn't pursue his work on showing chemistry was important for life because he lost his head. Here's another picture at the guillotine. You can see the guillotine here. He was a supporter of the French Revolution, but he unfortunately was also a tax collector with the old regime, and that led to him being tried in 1794 when hardly anybody survived. And his judge said that in giving judgment, when special pleading was made, here was a scientist who supported the revolution, could do much for the Republic. He said, "The Republic has no need of chemists and savants." Us scientists have to be careful when we mix with politicians, I would say. So this idea was picked up by Louis Pasteur, who went on to show that microbes were responsible, microbial cells, life for fermentation, showing that yeast was responsible for good fermentation. And he built on Lavoisier, concluding fermentation was a physiological act, and that these chemical reactions are an expression of the life of the cell. I emphasize all of that simply to show that it wasn't obvious that life was built on chemistry. It took a long time to do that. But when it was established, which is in the middle of the 19th century, then attention was paid to what sort of chemistry it was. Now, the basic building block of life is carbon, and for a while it was thought the sort of chemistry that might go on in living organisms might differ from that which could go on in a test tube. Then it was shown that you could do a synthesis of carbon-based compounds in the test tubes by Friedrich Wöhler, working with urea, also in the first part of the 19th century. So the chemistry of life was also the same as the chemistry of the natural world and was built on carbon. So what's special about carbon? Well, the first thing I'd like to say about carbon is that it's made in dying stars. That's where it comes from. It's the 14th most common element in the universe, and every one of us in this room, built on carbon, have our origins in the remains of dying stars. Extraordinary to recall that, but that's exactly where we've come from. Now, it has a number of features which are important for chemical diversity and therefore its special role in life. The first is that each carbon atom has 4 free electrons, so can form up to 4 covalent linkages. And you can see examples of that linkage on this slide here, where we have carbon atoms in grey. The second point to say is that the numbers of bonds can vary between carbon atoms. Carbon can form polymers, and that's extremely important. I'll say something more about that in a moment. The numbers of bonds can vary between carbon. You can get 1 or 2. You can vary the angle, which gives you different shapes to the molecules that will form. The third point is that carbon readily forms covalent bonds with other atoms, other elements, nitrogen and oxygen, and also hydrogen. And these lead to different chemical properties. It leads eventually to either positive or negative charge, acidic or basic, or the ability to repulse water. So there is a great deal of chemical versatility. Now, the carbon-carbon linkages, for example here, means that you can build up very, very long polymers. And that is absolutely crucial to understanding life, because it's important for encoding information, as we shall see a little later. You can translate a long polymer easily into information, which is crucial to life. So all life on Earth is based on carbon, and it's a very important characteristic. So if you were to send a spacecraft somewhere looking for life, a good starting point would be to look for complex carbon-based molecules, because they are associated, at least on Earth, with living organisms. And that's one way in which life has been looked for in space probes. Is it possible to have a different sort of chemistry underpinning life? Well, I certainly think it's possible to imagine a different sort of carbon chemistry, but it may also be possible to imagine another element apart from carbon. And the one that's usually discussed is silicon. It's adjacent to carbon in the periodic table. It also has 4 electrons. It's in its outer shell, but it's less reactive. There's a lot of it. There's the 8th most common element in the universe. And there is a proposal, which I shall discuss briefly later, that life forms could perhaps be based on silicates, particularly silicates in clay. And I will say something about that. So you can imagine life, and I'll show why, which isn't based on carbon. Now, there's a second. If we look at carbon, it can form these complex structures, as we can see here. As I've mentioned to you, you can get different covalent linkages that give different properties. These are amino acids here, so we get a positive charge here with lysine, for example, a negative charge here with aspartate, where we have oxygen, here we have nitrogen, and then side chains that repulse water. So this is what gives you the chemical variation. And here you see the ability, this is in a protein, by stringing together lots of amino acids to actually generate quite complex structures, which when they fold up will actually form 3-dimensional structures that are important for function in a way I shall show in a moment. Now, this structure, which is quite elaborate, is actually a single, of course, polypeptide, single chain of basic carbon-based building units, amino acids, and it creates a molecule of a particular shape with little mountains and little valleys, little clefts and gaps, which are then bounded by surfaces with certain chemical properties, so having positive charge, negative charge, or repulsing water. And this will be important in a moment when I talk about metabolism. So that's the first attribute. Carbon is the basis of life as we know it on Earth. Now, the second attribute, which is important for thinking about life, is that life has a metabolism and life can self-organize. And the second of these was emphasized by Schrödinger in his particular book. Now, metabolism is the ability to undertake chemical reactions in living organisms to sustain life, to build up molecules, anabolism, and to break down molecules, that's catabolism. And this is brought about, on the whole, by protein-based enzymes, not completely so, but on the whole, by protein-based enzymes, which are chains of amino acids. And I've shown you a chain of a protein a couple of slides ago. So enzymes play a critical role in metabolism with these specific structures that I just showed you. But what's really, and if you look at this, that these structures, these shapes, what's really interesting about this is that they form particular and very specific shapes that allow substrates to fit in and to be changed by breaking bonds and reforming bonds. And I'm demonstrating this to you, that here we have, this is a classic lock and key with the substrate looking like the key, the enzyme looking like the lock into which it fits. And if you have some alterations, and you can see there's a difference in the substrates here, it no longer fits, so it can no longer work. But the point about this is that this exquisite fitting allows you to break and reform covalent bonds based on carbon chemistry under very gentle conditions. And you can generate, by putting stringing enzymes together, a whole collection of different reactions that bring about the metabolism of the cell. But it's an unusual metabolism in the cell, an unusual chemistry, because many, many hundreds, indeed thousands, of chemical reactions are going on at the same time within a living organism or even within the same single cell. This may be familiar with you. This is a partial metabolic map. Each one of these dots in here represents a chemical transformation, a catalysis. This is actually the Krebs cycle in here. And you'll see there are hundreds of reactions on this metabolic map, which is only a part of the total metabolism of the cell. Now, what's wonderful about this is, of course, a characteristic of life, that it's all occurring at the same time in tiny objects, in cells often only a few micrometers across. And that is really quite extraordinary. For us to be able to carry out many of these reactions would require much more violent conditions, and we would be doing just a few reactions at a time. The cell can do it in a tiny space, and it can do many, many hundreds of reactions simultaneously. And I emphasize that because that, I suggest to you, is the way you need to think about cellular structure. This is a single cell from a living organism. And usually this is just described in terms of structure. There's a nucleus, there's mitochondria, there's different other organelles and clefts and nooks and crannies. I would like you to look at that and see this. That is to say, what this is generating is many, many micro-environments which can bring about chemistry of different sorts in different parts of the cell. So these are all microchemical environments that allow enzymes to generate metabolism throughout the cell simultaneously. And that is a different way of viewing structure within a cell, which is critical to life. So what we have here is a bounded entity of a life form. Here we see it in the level of a cell, which is undergoing metabolism, making molecules. And it's building up complexity at the expense of the order which is outside these boundaries. And that's another important aspect of life, because at first sight this seems to be in conflict with the second law of thermodynamics, and people get excited about that, but it is of course not in conflict with that, because this is local order, and that order can be generated as an outcome of disorder outside that bounded entity. So what we see in a living organism is a reverse of the second law of thermodynamics. We see a building up of complexity. Now, this metabolism within this bounded entity leads to another characteristic of life forms, this ability of creating order from a simpler, disordered surrounding environment. And this should be contrasted with some other life forms which are partial, or what I call sub-life forms. An example would be a virus, because a virus can only carry out its viral metabolism when it's embedded in an equally complex environment of another living organism. In other words, it's only got some characteristics of life. It can't do its own metabolism unless it's embedded in the metabolism of another living organism. Another comparison that I'll make here as well is that computer-based life, because sometimes people say, what's the difference between computer-based life, these games, called life, and they sort of do stuff on the computer screen. Computer-based life can only survive in the complex environment of the computer. They are not free living, maintaining their metabolism surrounded by a simpler environment. And that sort of helps working your way through this maze. Of course, we have parasites, which have to live in complex environments as well. So as with all of these things, it's difficult to find a single definition, but I hope that may actually help. Now, metabolism, returning back to metabolism, is the basis of generating higher order structure or organization, which is typical of life forms. If you think about it, all living organisms have particular shapes. I mean, we see it at its simplest level with this cell, for example, but we see it with our cells. We are all recognizably a human organism basically by our shape. You all recognize an oak tree by its shape, and so on. And forming structure is a key aspect of life. And you see it in its simplest form with simple molecular assemblies like this bacteriophage here, which is at a very, very tiny level, or a ribosome. And these are simply molecular assemblies based on direct atomic and molecular contact. In some ways, they're not very interesting, although I'm sure the crystallographers in the audience wouldn't agree with me, because they are simply built up on the simple laws of chemistry. More interesting for me is when you get into structures that are larger, like this is another picture of a cell, because now we have order across a larger domain. In the case of cells, it will range from 2 or 3 micrometers up to even 0.5 millimeter. And this cannot be generated, this order, by direct molecular and atomic contact. It has to be order that is produced at a distance. That's even within the cell, and of course in more complex organisms, and here this is a plant cell, and I couldn't resist putting up one of the very beautiful early pictures of a plant cell made by Nehemiah Grew in 1682, only a few years after Hooke described the first cells in Cork in 1665, or of course making animals, and here we have a mammalian embryo. So here we are seeing in life a spatial organization spread over a distance. And how that order is generated is one of the great problems of developmental biology. It certainly involves chemical gradients. It certainly may involve chemical waves, which will be important for this. And thinking about this has excited the theoretical biologists, and many speculations have been made that Alan Turing's, the great mathematician that produced work that underpinned the modern computer, also produced some equations showing how with the so-called reaction diffusion model, where you have reactions going on as chemicals diffuse through a space, and this is on the millimeter scale, and it produces order in the space. In this case, these concentric rings that you see here. And this is what I mean by spatial structure or organization at a distance. It relies on the dynamics of the chemical reactions, not on direct molecular or atomic interaction. So this metabolism is quite sophisticated. It can generate high-level order, and it is exquisitely regulated. I'm going to say a little more about regulation later, but by regulation, I mean the ability to control itself. And I'm going to just show you 2 slides about that. One to illustrate the principle. This is, if you go next door in the Science Museum, you'll see quite a lot of these, okay? This is actually a picture of a steam engine's governor that I took in New Zealand on a steamboat made in 1890. And one thing about engineering is, if you're like me, you understand all machines that were built before 1900 and nearly no machines built after 1900. And that's why I go back to a governor, because what we have here is that this is attached to the spindle being rotated by the steam engine. As it speeds up, the balls go out due to centrifugal force. We sort of get that. That pulls up this valve and shuts off the steam going into the engine. It's a classic negative feedback loop, and that's shown up here. This is one in a biological context where we have enzymes, for example, catalyzing A to B and then B to C, and the notion here is that as C builds up, then it inhibits the reaction from A to B, and as a consequence, the rate of the production of C goes down, and it's a homeostatic mechanism. It keeps C, ultimately, at a constant level. Very simple mechanism. It's the sort of thing that works in a thermostat that you might have in your home, except these days they're more intelligent than that, but that's another story. But this is an example of that negative feedback loop, as you see in the governor, or also in living forms as well. And I'm going to build a bit more on the concept of regulation towards the end of my talk. So what we have here is metabolism within the bounded entity, an extraordinary metabolism, which is different from, not in qualitative terms from the chemistry that we know, but just in its exquisite way that it can operate, which can lead to self-organization. I've emphasized self-organization over space, but a similar case can be made for organization in time. Living organisms undergo development in time, even from the simple cell. That's what I study, actually. The reproduction of a cell is a temporal organization in time, but as, of course, is our own development. Now, the next attribute, which is key for defining life, for thinking about life, is the other one that Schrödinger, in fact, spent most of his time on, and that is heredity. Heredity is really crucial because living organisms, it's a striking characteristic of all living organisms that when they reproduce, of course they have to reproduce, but when they reproduce, features that define what that organism looks like and how it behaves and how it works are passed on from generation to generation. It's one of the striking characteristics of life, one of the great mysteries, really, of life. And the basis of this was discovered by Grigor Mendel. I call him the great gardening genetic monk, because Mendel, this is when he was an abbot. He was abbot in Brno, although he did his work when he was just a monk, in the mid-19th century. Brno is in what is now the Czech Republic. He was trained as a physicist. That's important for the discovery he made, because he tackled this complex problem of heredity, which was of great interest, particularly to plant breeders at the time, but was really quite difficult. And he tackled it by choosing material he could understand. Now, that always sounds like cheating to most people who aren't scientists, but as Peter Medawar once said, science is the art of the soluble. You want to take on problems you can solve. It's no good having great thoughts about things that you don't actually solve. You have to choose problems that you can solve. Now, heredity was a great problem, but it was difficult. And the reason it was difficult is because genetics can be difficult. When many genes determine particular characters or important for difficult characters, they segregate and it becomes very complicated. So what he did was he tried out trial crosses with up to about half a dozen different plant species and chose to work on the P because he could make sense of the P. And he discarded the others. And that's very important. Scientists do that sort of thing all the time. They discard what they don't understand, they understand what they can understand, and then they can perhaps use what they've learnt to understand to tackle what they didn't understand. So it's a very important part. So he chose material that had clear characteristics like different coloured seeds or different heights and so on. It was P, and you can see here the different characteristics of the pea plants that he was interested in. And he recorded accurately, both qualitatively and quantitatively, what was going on. And that was crucial, as we shall see in a moment, and I'm sure reflects the fact that he was trained as a physicist. And then he generated theories and tested them, which also was influenced by his physical training. Having said all of that, he wasn't thought to be a very good physicist. And at university, he had to struggle to get through his physics classes. He was going to be a physics teacher, but I don't think they didn't think he was quite good enough. But I'm sure he learnt a lot from that. And he was set up in his monastery with actually quite good research facilities. We sort of often forget that our modern way of doing science is really quite modern. I mean, with universities equipped with good laboratories and so on, an invention primarily in Germany in the 19th century. But there were some monasteries which were very interested in science, and that was the case here in Brno. So he had a garden here, quite a big garden. I visited this. This is a picture I think I took. And next to it, he had a big greenhouse. It's no longer there. And the greenhouse was certainly bigger than this stage. And this was put up by the monks to allow him to investigate his plant breeding. This is a big facility. You know, I tried to put up a greenhouse, research greenhouse in Rockefeller of this size, and I couldn't afford it. Okay, and they're expensive. They're expensive facilities. So he was really quite well supported. He did do some physics work as well, actually. Since I'm rambling on a little bit here, I've seen his telescope. He made a map of the moon. And he also studied meteorology. So in a storm, he showed air pressure drop precipitously when you had a cyclone going through. But back to genetics. What he showed was that if you took, in his pea crosses, if you took two plants and crossed them, and that one would have, for example, green and yellow seeds or something of that sort, I think I have something of that sort. Then what would happen is when they were crossed, then one of the characters, green in this case, on the right here, disappeared in the first generation. But when you crossed that first generation to itself, the green reappeared, and it reappeared in a very precise ratio of 3:1. And that was really crucial for his thinking, because he thought this told him that it was like sorting out of combinations. And it made him start thinking of particles being segregated. And it was that theoretical leap that was crucial. You might be surprised to hear that Charles Darwin, in 1868, only 3 years after 1865, did, without, we think, knowing about this, also crossed snapdragons and observed the same 3:1 ratio. He counts it. It's in one of his books. But he's more of a naturalist. He just leaves it there as an observation. He doesn't actually make a hypothesis and try and test it, as Mendel did. But the 3:1 ratio was also discovered by Darwin. So what Mendel did was to propose a particulate theory of heredity, which underpins this discussion about heredity, proposing that there are unchanging particles, which we would now call genes, that influence particular characters and allow you to understand differences between characters in different organisms. That in each plant there were two particles that were important for the character that he was interested in, but only one was passed on to gametes. And on fertilization of pollen with an egg, then you got those two single particles back together again, so you had two copies. We'd now call that diploid. And then finally he proposed that one particle might dominate in its expression over the other. And that was an absolutely brilliant piece of abstract reasoning. I have to say nobody took any notice of it at the time, and it had to be rediscovered later, 35 years later. I don't quite know why nobody took any notice of it. It may have something to do with the fact that chromosomes were discovered in between 1865 and the early 1900s, and then you began to see a sort of concrete basis for the abstract reasoning. And in fact, chromosomes were seen to change in number when undergoing gamete formation, which is exactly what was predicted. So implicit, incidentally, in Mendel, though he doesn't say it, is that these particles encode information. That's absolutely implicit. It doesn't make any sense unless you conclude that. So a few years later, chromosomes were discovered and were thought to be the bearers of hereditary information. And later, in the 1940s, Avery, McLeod, and McCarty discovered deoxyribonucleic acid, DNA, as the basis of heredity. That was done at Rockefeller. I have to tell you, when I went there in about 7 or 8 years ago, I was being introduced to everybody, and this little old man came up to me and shook my hand, and he said his name was Mac, and I said, "I'm very pleased to meet you, Mac." And then he said, "Mac McCarthy," and I sort of looked at him and I said, "You can't possibly be the Mac McCarthy." And he said, "Oh yes, I am the Mac McCarthy." And why I was so astonished is, of course, I'd been taught this in the 1960s, and I already thought he was dead already, because this had been published in 1944. And here he was in 2003, still going to seminars at Rockefeller. Anyway, through pneumococcus transformation, they discovered DNA was the hereditary material. One of the great discoveries of biology of the 20th century, not rewarded with a Nobel Prize, one of the big mistakes the Swedes did. But of course, they did give it to the structure of DNA, which was solved by Crick and Watson based on data from Wilkins and Franklin. Both, as I've said already, Crick and Watson were influenced by Schrödinger's book, What is Life, and by Avery showing that DNA was the hereditary material. And you have the beautiful double helix, which is the basis of heredity. And then Crick went on to show, with others, to crack the genetic code, to establish the central dogma, where DNA encodes information in the polymer, the order of the polymer. It's transcribed into RNA, also in the order of the polymer. The specificity being given by the nucleotides that make up the polymer. Otherwise, it looks actually a very similar structure. But then the translation of that into amino acids to make the polypeptide, the protein, allows an extraordinary chemical diversity. And what's actually beautiful about this, in my view, is that the polymer chemistry that is needed to store genetic information is in the very stable DNA molecule, which is also not very varied in actual fact. All the variation is on the inside. The variation is only really in the order of the nucleotides. But then when the time you come down to the amino acid based polypeptide with all these different side chains, not only are there 20 amino acids as against 4 bases, but these can now wrap up into all these complex structures. So the information encoded here in a molecule good for encoding information is translated into structures that have the great variety of life, the chemistry of life that is actually important. So heredity is crucial for life. All living things have to have a heredity. In life on our Earth, it's based upon nucleic acids. There is some organisms with RNA viruses, and this beautiful explanation from the 60s and 70s of the central dogma of molecular biology. Now, heredity underpins the next characteristic of life, which is probably the most beautiful idea in biology, and that is evolution by natural selection. You knew it had to be up there sometime. Well, now you have it, okay? Evolution by natural selection, which is, of course, based upon heredity. Now, normally, we associate evolution by natural selection with Charles Darwin. But there's two aspects, of course, to this. Evolution, that is life forms evolve, and secondly, the major mechanism, which is by natural selection. And the first of those, that life evolves, is absolutely not Charles' idea. It was discussed widely in the 18th century, in particular by his grandfather, the irascible Erasmus Darwin. And I just want to say a few words about Erasmus Darwin, because he is, frankly, a much more interesting character than Charles. And people don't know about him, and he's one of the great British scientists. I have to mention, in fairness and openness and glasnost and all that sort of thing, that the French also contributed greatly at this stage too, particularly Buffon and Lamarck. But we're going to focus on Erasmus. This is Erasmus Darwin. He was a doctor in Lichfield and Derby. He was very fat, a great conversationalist, although stuttered all the time. He was so fat that he had to cut out an oval from his dining room table so he could sit nearer to his food. Like most doctors at the time, he wasn't actually terribly good at curing you, but he was very good at telling you if you were going to die or not, which was extremely useful, I think, because it means you could set your house in order and write that will that perhaps you didn't want to write until the last moment because you didn't want to upset your relatives. He believed in evolution, and he had a motto on his coach in Latin, which translated says, everything from shells. And what he was thinking of, if you open up a shell, the mollusk is completely formless inside, and he was arguing all life as we now know it came from formless objects like that. The Dean of Lichfield didn't like this, and began to tell his richer patients they shouldn't go to him, so he had to paint it out so he could keep his patients. He was also, incidentally, a poet, and he wrote up most of his science in blank verse. So you get these I've got a number of his early books, actually, from the 18th century, and you read these ludicrous poems that are all talking about the sex life of plants and minerals and all sorts of other things, all written up in blank verse. He was a proponent of female education. He was a founder member of the Lunar Society, who used to meet under the full moon, including characters like James Watt and Wedgwood and so on. So a very, very interesting person. And the idea that life evolved was clearly being discussed and believed by many scientists. But they didn't have great amount of data, and that was the point where Charles Darwin, looking rather serious over here, grandson, and didn't really acknowledge Erasmus as perhaps as much as he should have done, quite frankly. And he during his voyage of the Beagle, amassed a huge amount of data, both for the fossil record and observing life, to support the idea that life had evolved. And that's another point about science. It really isn't enough just to have a good idea. You also have to have good data, observation and experiment to support that idea. And that's where what Charles had completely in spades. Now, the second, and so Charles wrote this up in The Origin of Species, 1859, and he has one illustration in that book, and that has a tree of life in it, as you can see here. He doesn't seem to be too clear whether life originated more than once, if you look at the bottom here, but absolutely life is related and connected to each other. Incidentally, the second half of what he had to say, which was natural selection, also wasn't solely Charles's ideas. I shall tell you in a moment when I explain about natural selection. Now, his idea, which he beautifully expressed, was that a major driver in evolution is due to natural selection, and that comes about when you have inheritable variation in populations due to genes, that when variants are successful within that population, those particular organisms reproduce more, and they pass on the particular gene variants that lead to that success, and that these changes over time can lead to speciation. That's the idea of natural selection. But it was the same idea occurred to Alfred Wallace in 1858, and he sent, by one of these curious coincidences, a letter with a paper he wanted Charles to communicate to the Linnaean Society with exactly that idea. Charles had been sitting on this idea for 20 years. He didn't know what to do. It's a story I'm sure many of you know. Hooker, who was then director of Kew, suggested that Darwin write a paper, and they both submitted it to the Linnaean Society. It was in Burlington House in the Royal Academy. If you go there, you'll find the Darwin and Wallace room where they read it out. And once again, nobody took much notice of it. They did take a lot of notice of the book in 1859. Once he did publish it, though, a certain Patrick Matthew, who had also had the idea of natural selection, had written it in a book on naval timber that he published in 1831, and he sent it to Charles, and Charles acknowledged it in later editions. It was only one page, but the idea was there. Now, the idea of natural selection, I think you've got it. This is based on the Galapagos finches that Charles studied. This is good for cracking nuts. This beak is good for finding insects. And here, we do it with pliers, and we are selected to do their job, just like in case of the beaks. Now, I just want to point out one thing to you, is that there are certain characteristics that living things have, which means they will undergo evolution by natural selection. This is a single-celled life, and those properties are that cells reproduce, living organisms reproduce. If they have a hereditary system, shown by these little DNA molecules in here, which are passing on information, and if that hereditary system shows variation, either due to mutation or problems with copying, then as a consequence of that, you will get a variety of forms generated, and if some of these can reproduce better, then they will take over the population. So if, for example, red shells are better than orange ones, that will eventually dominate. That means if you have a hereditary system set up in this way, then you'll get evolution. You'll get evolution by natural selection. They are completely linked. The idea of a cell, the idea of a hereditary system, and evolution by natural selection. To the point that Muller, and this I really would like you to pay attention to, defined life in quite a different way. Not how it looks like or what's happening into it. He said living things have properties which allow them to undergo natural selection and therefore to evolve. It's like a historical definition of life as against a description of the chemistry or the function. And that means that you could imagine different life forms, completely different to the ones that we imagine, which have these features that I've described here, but are not based on carbon or anything that we know. And a very imaginative Glaswegian chemist called Graham Cairn Smith, in the 1970s and 1980s, and I heard him speak in Scotland in the 70s, imagined a primitive life form built on clay particles, on silicates. And what he was imagining there is that different minerals embedded in the clay could affect the properties of clay. So that if the clay was sticky, then more silicates would bind to the clay and it would grow faster. And if it had other minerals embedded in there which allowed the clay particle to fragment, that meant it would reproduce faster. And so you could actually generate a primitive hereditary system based upon the pattern of minerals embedded within the clay, and you could select for clay particles that grew faster and reproduced faster. And he postulated this as a primitive idea of evolution by natural selection. And I only mention that is because maybe there are life forms here built on some different structures, different ways of doing it, or perhaps deep in the earth, or perhaps on Titan, which are completely different to life forms as we understand it. Because if it's evolution by natural selection, it doesn't have to be built on the same chemistry as we've had. Now, I'm getting, you'll be pleased to hear, as it's 7 o'clock, to the very last few minutes of my talk. Because throughout my lecture, there's been a recurring theme. I kept mentioning information in a variety of spaces. And another way of viewing a living organism is that it's a complex system. Now, of course, there's many things that are complex systems, but once again, this is exquisite. So I want to turn to the final point. Oh, this is Darwin's beauty. Can I just quote Darwin's last sentence in Origin of Species, because the last word on him. Whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful have been and are being evolved. I wish I could write papers like that. I wish I really, really could. And furthermore, I wish those damn editors would accept it if I did. Because I tell you they wouldn't. Anyway, that's Charles. Systems and information management, crucial for understanding life. And I want to end my talk with just a few thoughts about that. Because the chemical and physical processes of life, the metabolism, the self-organized entities, which I've been talking about, are necessary to generate higher order properties of life: homeostasis, repair, regeneration, reproduction, the ability to communicate between living organisms, and all the ability to have a hereditary system. And these higher order biological phenomena are a consequence of having an effective working complex system. And we have to think about complex systems more, particularly how they manage information, how they gather information, store information, process it, how they reproduce that information. And this management of information is embedded in a highly regulated system of networks. And these will exhibit behaviors in such a way that, in my view, living organisms only really make sense often when they're described in terms of information and the management of information. Now, I'm going to give two examples just to demonstrate quite why I'm so enthusiastic about this. One is, we've already talked about it, DNA. DNA is a beautiful structure. We understand the chemistry. It's a double helix. We understand that's all crucial. But it only makes sense for biology when you recognize that this is a digital information storage device. You have information there stored by the nucleotides. So the structure makes biological sense when you translate it into a concept of information. That's DNA. If we now look at this slide, I showed it to you before. This was discussed first of all by Jacob and Monod in regulation of the lac operon, and they postulated that the lac operon had a negative feedback loop, and that this negative feedback loop determined the rate at which the lac operon was transcribed. Once again, you can describe it entirely in terms of the chemistry. You can describe it in terms of protein touching DNA and proteins touching sugars and so on. All that would be is a chemical description. It all makes sense, it comes alive, it's important for biology when you realize it makes a negative feedback loop. That is also how you manage information. So by putting together these sorts of circuits, all sorts of things can emerge. You can make switches. I put a positive feedback down here, which is a switch, timers, oscillators, toggles, and you get a good metaphor of all of this if you take an electronic circuit. This is the inside of a radio, which I stole from a journal where somebody had plastered all these genes on it to show that these genes are interacting with each other in forming this complex network. But as Dennis Bray has emphasized, living organisms, though you can use them as a metaphor based on the hardware of an electronic circuit, life is actually what he terms nicely wetware, where the wires that connect the different components are actually diffusible chemicals. And what that means, of course, is that you can very easily rewire. You can connect different components because they're diffusible. And this is incredibly rich. And I don't think we thought closely enough about that, because this is much greater versatility than a computer, where you have a certain hardware and then you put the software in. What we're doing here is you have potential different software, and that's the usual metaphor for life, but we can also play with the hardware much more readily because it's actually basically wetware. Now, if we take this type of approach and think about systems, we can learn about living organisms by looking at other complex systems, and that's why I mention it to you. And very briefly, transport hubs, where we have an interactome essentially here, and it's organized in a scalar fashion which bears some resemblance to how components interact in living organisms. What it tells you is that certain hubs, both in living organisms and also in aeroplane routes, are much more important. This is Houston, I think. You take out Houston, the whole system falls down, and nobody cares at all about Kansas City, which is somewhere over here. Quite right too, really. But the second thing we have to understand about networks is that although we would love them to look like this, you know, and that's how our brains try and force living organisms, they are much more likely to look like this due to evolution by natural selection, a final point I make. The networks are complex. They are not, in fact, intelligently designed, you know, to be a minimal system. They are put together. Here you know exactly what happens. Speed that up or slow it down, that's what happens here. Here, you know, if you eliminate this, what happens here? I don't know. You know, you can't work it out. It's not naturally intuitive. But we usually think like this, but we have to think like this. Now, this is usually associated with only some of us, and I take this picture from a pilot's manual handbook, and I don't dare show this in the US, okay? And men think simply and you're on or off, you know, and women are a little more complicated, okay? And they think in more complicated ways, and as a consequence, they can cope with this much better. We have to think much more like this in dealing with the complex networks of living organisms. Another point about this, which is why I think complexity is going to be so important in biology, is that if you do have a simple linear pathway, then if you exploit the dynamics of that pathway, then you can convey much more information. So pulsing information down here can mean at different frequencies, you could convey quite a different message here with that pattern versus that pattern. And that seems trivial, but it really, really, really isn't, because let's go back to a metaphor. If you just have an on-off signal, you can just control a traffic light. If you have dynamics, you can generate the Morse code and you can write the works of Shakespeare. This is a metaphor. I'm not suggesting that, of course, you can write all those words with living things, but I am sure, I'm utterly convinced that life exploits dynamics in its pathways much, much more than we've ever thought before. And all of this is going to be built into life, and I think characteristic of life, even if we don't yet understand it. But one final point to make about this. This is not intelligently designed. And to show something that is intelligent designed, I show you Harrison's clocks. He was the guy who invented, in the 18th century, the very accurate chronometer. He made this. They tested it. It didn't work quite accurately enough. He went back to the drawing board. He made that. It didn't work quite well enough. He made that. And he eventually ended up there. This has no remnants of that in it, whereas living things have the remnants of everything that went before it. And that's why it's so difficult to work out, because we have redundancy and complexity, and it's not how we would simply actually make something. Now, if we have this complex world, I think it's possible that biological thinking, and therefore insight as to what is life, may move us from what we tend to have today, which is a common sense view of the world in biology. I inhabit a common sense world. I imagine when I do my cell biology, the cell in a large space, and I've got a molecule over there touching this molecule here. And you sort of build up pictures in your head of that sort. Going back to the physicists, most of physics prior to 1900 was also in the common sense world. That's where Newton works. You get to 1900 and we get relativity with Einstein. Now, that's one of these ideas that you read it, you think you've got it, you close the book, you put it away, and it slips away, you know? Somehow it doesn't hang in there because it's not commonsensical to sit on a beam of light traveling at the speed of light, or even now faster than the speed of light. And you just cannot imagine. It's not part of the common sense world. But then you go 25 years later and you encounter Schrödinger and his friends, and you're in complete Alice in Wonderland when the cat can be alive and dead at the same time. This is not a common sense world, and I do wonder whether the complexity I'm talking about will take biologists into a less commonsensical world, and we may need the more abstract thinking that physicists are used to in dealing with their subject to make sense with what we are doing. So this area of complexity, I think, may have many consequences. What is life? I haven't given you a definition of life, but I have listed some attributes that I think are important, that I think are also interesting in thinking about life. There is a particular chemistry. It's based upon carbon, but maybe other life forms could be based on different chemistries. We will have metabolism, homeostasis, and self-organization. That gives you the beauty of life, the complexity of what we have when we look around us at all living things. That's underpinned by heredity, and that heredity can lead to evolution by natural selection. So life is not static. It can change with the environment, and it can generate the wide and great diversity of living organisms. And key, I think, to understanding great chunks of this is if we focus on systems and the management of information. But trying to define life, trying to actually say which of these characteristics is more difficult. Because if we take a virus, it will only have aspects of this, particularly aspects which are concerned with whether life is free living or is bounded by a bounded entity which beyond it has got a simpler environment, and it has to build up from that. Now, I want to finish with one other thought. I think it is possible that in the coming decades something may happen that will really sharpen our thinking about what is life. And that would be the discovery of a new and different life form. That could come from space probes. It could come from Titan, as I've measured, the satellite going around Saturn, which has got water, or thought to have water on its surface. It could come, I think, by burying deep into the earth. There may be other forms of life that we've not thought of. I could easily imagine life built on a different carbon chemistry, or not easily, but I could imagine it. But maybe even more entertaining would be ideas such as K and Smith, which is life forms built on a different principle. And that simply may be difficult to recognize. And I just wonder, is it there already? We just don't know what we're looking for. Maybe we'll find that in the coming decades. Thank you very much. Thank you.